Hey guys, welcome back. We're gonna do our second lecture here over cells, and we have just finished up prokaryotic cells, where we kept things pretty simple. We really only had about 4 or 5 things that were super important with prokaryotes, and we're gonna move on to eukaryotes, which is you and me, and they are made of multiple cells generally. We do have a few that are just one cell, but their cells are gonna be quite a bit more complicated than the prokaryotic cell was. So let's jump right into it. Eukaryotic cells are going to have a nucleus, and the nucleus's primary function is to contain the DNA, to keep that DNA safe because it's pretty valuable cargo. They are going to contain other membrane-bound organelles, and we're going to learn about a lot of those here in just a minute. Membranes allow our organelles to have a higher degree of regulation on what's coming into the organelle, what's going out of the organelle, and that's a really important feature. And they also have specialized environments that allow the organelle to have a particular function. We call this compartmentalization. We have one compartment of our cell that does this, and we have one compartment of our cell that does this job. It's just like a big company that has different departments, and if you call them, they might transfer your call to a different department because they have certain departments that handle certain things, and your cell is just like that. And so it's going to make your cell a lot more efficient, having those compartments where it can keep certain jobs separate from each other. So some organelles that you should be familiar with. If you're not already familiar with them, I would like you to be really familiar with them here in the next 30 minutes, and here they are. A nucleus, and you know what that does. It has the DNA in it. The endoplasmic reticulum, it's going to make proteins, it's going to make lipids, do a few other things. The Golgi is going to modify and sort these proteins and lipids. Our vesicles are going to transport substances or store substances. Mitochondria, you probably learned this as the powerhouse of the cell when you were younger, and this makes ATP by breaking down glucose. Chloroplasts, plants, not us, are going to make sugars, and lysosomes are going to digest things. Peroxisomes break down toxins. Vacuoles are generally storing water, but they can store some other things. And then organelles here, we have a few without membranes. Ribosomes do not have membranes around them, and they assemble proteins. And a centriole does not have a membrane around it, but it anchors cytoskeleton fibers. And then we have our cytoskeleton, which we'll get into in the next lecture. We won't worry about it too much just yet. So here it is in all of its wonderful glory, a big, complicated plant cell. But here's all the stuff that's in it. Each has its own job, and each is very important. The thing to note right away when you look at plant cells as opposed to animal cells, let me see if I can get you an animal cell. Here's an animal cell. Okay, so you look at the animal cell, you look at the plant cell. Very, very different, okay? The first difference I want you to note between these two is plant cells have a very large central vacuole. And this here is actually quite a bit smaller than they can be. A lot of times, they can take up easily 80 to 90% of the cell and just shove all this other stuff right in here just against the cell wall. But you're going to see all sorts of structures in here that are all working together. We have our central vacuole. It's full of water, salts, dissolved ions, that kind of stuff. We have our nucleus. It's got a nuclear envelope around the outside. It's got DNA on the inside. We have ribosomes all over the outside that are assembling little proteins. We have a rough ER assembling proteins. We have a smooth ER. We have a Golgi body packaging and modifying proteins. We have a vesicle down here. Not really sure what it's doing. They're saying lysosome, some sort of digestion. We have a plasma membrane to keep the outside of the cell on the outside and the inside of the cell on the inside. We have a cell wall outside of the plasma membrane for additional support. We have chloroplasts that are filled with these little thylakoid discs that have chlorophyll in them, and they're capturing sunlight and turning it into sugar. Then the opposite, this guy over here, the mitochondria, is taking the sugar and burning it to make ATP. So these guys are kind of doing the opposite jobs. Plasmodesmata, so we can easily throw sugars and waters from this cell to his neighbor and from his neighbor to him. And then little cytoskeleton fibers that are supporting these structures, growing new cell walls, that kind of stuff. And it's all working together at the same time. It's kind of an amazing thing. So that's what's in a plant cell. Here's an animal cell. You take away the cell wall, you take away the central vacuole, and there's a lot of similarities. So let's look at the animal cell. It's a little easier. The first structure they're giving us is the rough ER. They call it rough because it has this grainy appearance. That grainy appearance is due to ribosomes. And so when something's covered in ribosomes, and ribosomes serve the function of making proteins, it's a pretty safe assumption to say this thing probably makes proteins, and you'd be absolutely right. So it's going to make and modify proteins with all these ribosomes that are attached to the rough ER. Now next to it is the smooth ER, and they call it smooth because there's no ribosomes on it. It'll make lipids, it'll break down carbs, it'll detox certain substances, and so smooth ER does a very different job from rough ER, but they're both very important. We have the Golgi body. It looks a little like the smooth ER, just a little less like coral reef and a little more like messed up pancakes, and you're at Golgi body. It's going to take the products that this rough ER has made, sort them, modify them, attach carb groups, cut proteins in half, that kind of stuff. Package them for export sometimes if there's something that the cell's going to get rid of. They can bud off the Golgi into a vesicle, and that little vesicle can be transported to the plasma membrane, fuse with the membrane like you see here, and then just expel its contents out into the extracellular matrix for whatever its contents are going to do. So down here you have a lysosome. A lysosome is a little vesicle, and they're all over the place in here. And this little vesicle is going to digest and recycle cellular debris, because cells are just like you and I. They make waste, and they've got to get rid of the waste. Just they don't cart it off to the dump and put it in a big hole in the earth. They've got to break it down right there in-house at the cell, and they've got to recycle everything that they possibly can and then break down any harmful wastes. And so we use a lysosome for that. And so there's your animal cell. The rough, the smooth ERs, the Golgi body, and the lysosome. Not too bad for a little introduction to the endomembrane system. So we're going to focus now just on the nucleus. So if we go into the nucleus, which we've avoided so far, eukaryotic nucleus is going to contain all the DNA, and it's going to keep it from any other damage that could go on in that cell, because we can rebuild a lot if we've got the information to do it. If we damage the information, we're in a lot of trouble. Chromatin is a term that we use for DNA when it's all wound up with its associated histone proteins. Without being really wound up, DNA would be super, super, super long. And so the times that we see DNA, really it's in chromatin. And then we have a nucleoplasm, which is the fluid interior of the nucleus, just like the inside fluid material of our cell. We have a nuclear envelope, and the nuclear envelope is a double membrane with nuclear pores, and it does the same job our cell membrane does. It keeps the outside out and the inside in, and it lets certain things go out and certain things come in. And then the cool thing about a nucleus is inside that nuclear envelope we have a nucleolus, and it's this really dense region of proteins and nucleic acids. And what the nucleolus really does is it builds ribosome subunits. And so ribosomes have a small subunit and a big subunit, and we will build those inside the nucleus and then shoot those little guys out into the world. And the small ribosome unit and the big ribosome unit will come together, and they can start making proteins for us. And so we are really focusing on information and protein building when we're in the nucleus. Those are some important structures in there. So here's the picture. Super hard to see what in the world's going on there. Here's the cartoon. Not so bad. Okay, so you can see we've come. I like the animations a little better than this guy, but it is a cool example. Scanning electron microscopy, right? We add the color to make people think we can take color photos. Not true. And then transmission electron microscopy. We cut that nucleus in half and see what his insides look like, right? Very different approaches, but both are valuable. So nuclear envelope, it's on the outside, keeps the nucleus protected. Nucleoplasm, fluid on the inside, fluid on the inside. Chromatin is the DNA strands. Chromatin is DNA strands in here. Our nucleolus is a super dense region, and right here, it's a super dense region right there. And then our nuclear pore lets those little strands of, messenger RNA out. And here's your nuclear pore right here as well. So there's the nucleus. Now we're going to switch gears here to the endomembrane system. So endomembrane just means within a membrane. It's a pretty general term there, but it's basically all the organelles in the cell that are between the nucleus and the plasma membrane. So all of them. It makes, modifies, and transports proteins. We focus a lot on proteins in the cell because proteins do a lot in the cell, but we'll also make lipids and transport lipids. And we're going to secrete those guys, or we'll put them in our cell membrane, or they'll go do a job somewhere else in the body, something like that. The other thing we're going to do with endomembrane systems is destroy toxins in the cell, recycle our waste materials, and a few other specialized functions as well. So here's our endomembrane system for our animal cell and our plant cell. We've talked about a central vacuole a little bit. It's a glorified water bag. We've talked about rough ER already, building proteins. A smooth ER is our little coral reef that will, work with lipids and fats and detoxify stuff. Our Golgi body, which is going to be over here, this little flattened pancake thing and flattened pancake thing here, which is going to package proteins and transport them. And then finally our vesicles down here. And there's a couple types of those we'll talk about. Sorry about these lines getting all messed up, by the way. That's kind of tricky to read. So here's the ER. Endoplasmic reticulum, the ER, an extension of the nuclear envelope that forms a continuous folded compartment. So all that means is it's a bunch of flattened sacs together. Two kinds, the rough ER, which we've talked about, are going to fold up our polypeptides of proteins into their tertiary form. Proteins have four forms: primary, secondary, tertiary, and quaternary. Quaternary is the finished product, fully functional protein that's ready to go. The rough ER is going to get these guys to tertiary, which is pretty close to ready to go. So that's an important job. And then our smooth ER is going to make lipids, and it's going to break down carbs and other lipids and detoxify poisons. So both very important: building proteins, getting rid of poisons. All good stuff. So vesicles. There's a lot of different types of vesicles, and they all do different things. But really just think a tiny clear sac in the cell is what a vesicle really is. They store or transport substances. There's two types I want you to know about. There's peroxisomes, and peroxisomes are little vesicles that contain enzymes that'll break down basically poisons. Hydrogen peroxide, not a good thing for a cell. Alcohol, not a good thing. Other toxins, peroxisomes are going to break those guys down, so they're not going to do damage in the cell. Then there's lysosomes, and these guys are going to do a similar job to peroxisomes, but they don't specialize in poisons, like think peroxisome P for poison. Lysosomes are going to digest waste. So just your regular old metabolic waste that you make from just being alive, that's what these guys are going to digest and get rid of. So our vesicles, they have a few different functions. Some of them will just dispose of waste, debris, or toxins. And some of them are larger vacuoles like we see in plants. Here in a plant, you could have a vacuole that's anywhere from 50 to 90% of the interior of the cell. So pretty large. And they store amino acids and sugars and ions and wastes and toxins. They're just this huge water bag in the middle of the cell, and they can dump or store whatever they want in there for a plant cell. The pressure from that fluid in the central vacuole is going to keep a plant cell turgid or firm. Basically, that's the reason your plants shouldn't wilt. If they get dried out and wilt, it's because they don't have as much water in that central vacuole. So that's a vacuole, not that fancy. Let's move on to Golgi bodies. Golgi bodies are a little more fancy. So folded membranes, they look like the flattened kind of pancakes, and they will finish off these proteins and lipids that are delivered from the endoplasmic reticulums to the Golgi, and they'll package and deliver these things to where they need to be. And some of these proteins might be just right there in the cell, and some of them might be heading out to the plasma membrane, and some of them might be excreted into extracellular matrix. They've all got different fates, and the Golgi bodies kind of are our mailman getting them there pretty much for where they need to be. So here's our interactions in the endomembrane system. So our vesicles, they bud from the endomembrane system, and they transport to the ER. Okay, when we get to the ER, we are going to make proteins, synthesize these proteins, and the proteins are getting the structure where they're going to start to become almost functional. We go to the smoothie yard, where proteins migrate through the interior and end up in the smoothie yard. Some of them hang out here, some of them move on, and the ones that move on bud off in a vesicle and head for the Golgi. Golgi takes the vesicle, they wind through the flat labyrinth of messed up pancake land, and poof, they come out the other side, and they are tertiary form, ready to rock and roll proteins, send us somewhere to do an awesome job, and then they're heading for the plasma membrane where they can, I don't know, communicate with other cells as cellular adhesion molecules so that this cell knows who his neighbors are. And that's the story of the endomembrane system. Crazy, huh? It's a lot of ground to cover just to make a protein. But it's a crazy world we live in. So let's move on to mitochondria, the powerhouse of the cell. That's what everybody knows, Matt, right? Eukaryotic organelle that makes the energy molecule adenosine triphosphate, ATP, through aerobic respiration. Aerobic respiration means it needs oxygen. We need to be breathing for this. It contains a couple membranes, two of them forming an inner and an outer membrane, and we build up all these hydrogen ions in that space between the inner membrane and the outer membrane, and all those hydrogen ions work as the fuel to turn a really cool protein called ATP synthase, and he makes adenosine triphosphate out of adenosine diphosphate and an inorganic phosphate molecule. So they make fuel. That's what they do. They take sugar and they turn it into usable chemical energy for everything in our bodies. And it's awesome. And they do the same thing for plants, and it's still awesome for plants. So we're big fans of mitochondria in the land of, things that want to survive. So that's us. It's got its own DNA. Weird, huh? Mitochondria has its own DNA, has its own ribosomes, and it can divide on its own. It doesn't have to wait for the cell to divide. It can just cut itself in half and divide. So it's weird. They're super cool. We'll talk about them more in class and endosymbiosis a little bit, which is fun. They resemble bacteria a little bit. I'll kind of let you be the judge of that. And the idea for endosymbiosis or endosymbiont theory is there's a potential for them to have evolved from aerobic bacteria that got eaten by a cell, but the cell didn't eat it. He's like, let's just be roommates and you can make energy for me. And then they lived happily ever after, and we'll discuss it. And I'll let you be the judge at the end of the day, but it's neat, at the least it's a neat idea. So that's mitochondria. Here they are, a little less than beautiful. Here's the mitochondria. So we got two membranes, one on the outside, one on the inside. We have these really convoluted interior, which AP biology kids helps us with what? Hopefully your brain is telling you surface area. When you see stuff like this, you think we have a giant surface area, and you would be thinking correctly. So the mitochondria has this outer membrane, this inner membrane, and then these folds called cristae on the inside. And these cristae give us this giant surface area where we have more room for more ATP synthase pumps, so we can make more ATP in one mitochondria. So we have this huge surface area to get this job done. But their structure is pretty easy. Inner compartment, inner membrane, outer membrane. That's it. I mean, they're really super basic. Throw a little DNA in there and you're in great shape. That's what you got for mitochondria. So, plastids. We got to leave you and I alone now. We're plastids, so we got to think plants, not animals or humans or fish. Plastids. So, plastids, there are many different types. You probably know chloroplasts, and that's about it, unless you've taken botany. So plastids are organelles that function in photosynthesis or not photosynthesis. They can store pigments, and they can store starches, and they can store whatever a plant needs them to. But we're only going to find them in plants and algae. So we have chromoplasts. Chromo means color, by the way. Amyloplasts, which is storage of starch, and chloroplasts. So chloroplasts are the ones that I want you guys to know the most about, and these are plastids that are specifically used for photosynthesis, which plants capture sunlight, turn sunlight into energy. That is photosynthesis. And the reason most plants are mostly green is they have a lot of chloroplasts filled with a lot of chlorophyll to make a lot of sugar from sunlight, and that's pretty much what they're doing. Chloroplasts resemble photosynthetic bacteria, something we'll talk about again. Is it another case for endosymbiosis? I'll let you be the judge, but they do have their own DNA, and they do divide on their own, so maybe. So here it is, the chloroplasts. You got outer membranes, 2 of them, of course. We have stroma, which is just fluid. It's really nothing that fancy. It's a fluid media for these guys to exist in. And then we have the inner membrane, which are called grana, or made up of thylakoid discs. And these guys are going to provide a huge surface area, right? I know you knew it. I know you did, for photosynthesis. The more surface area we get by stacking these goofy little pennies, the more sugar we make out of sunlight, and the plant is happy now. So that's where we're going to end for this time, because I don't want you falling asleep at your laptop. Take a little break, soak it in, look over your notes, look back over stuff you need to look at, and we'll jump back into the cytoskeleton next time. So thanks, guys.